Everyone, thank you for joining us. This is the Headwaters Head Royce School Program Experience. We are going live right now to share our student presentations. We're so excited to have you here. These are all of our high school students from Head Royce. It's been such a joy to work with them over the past 2 weeks. These are all high school seniors, so please, we do ask for questions from the audience, so please be sure to share any questions or comments you have on whatever platform you are watching this from, whether that be Facebook, LinkedIn, YouTube, or StreamYard itself. We're so excited to have you here. My name is Courtney Kudra, and I am the Research Experience Program Manager here at Headwaters. I've had the honor of working with this crew for the past 2 weeks, along with Morgan and Katie, who you will also see here, who are also program managers here at Headwaters. We're so excited to share this all with you today. So we're going to go ahead and bring up our first crew here. So we have their title of their slides is Our Soil Mate, and I'm going to hand it over to Morgan to introduce this group that she was able to work with during our field day.
Hi, everyone. I have the pleasure of introducing Sasha, Malcolm, Belle, Anoki, and Finch. They were really wonderful to work with last week. They worked really hard to collect some awesome data on soil and plant coverage around the lagoon, and I am very excited to learn about what they found.
All right. Hi, happy Thursday. This is our presentation for ESH, and we decided to research what the effect of soil moisture was on plant coverage.
I'm Finch.
My name is Malcolm.
My name is Anoki.
I'm Belle.
I'm Sasha.
And this is our research mentor, Morgan.
Okay, so we're going to go over first some scientific background, then our experiment, including hypothesis and methods, and results and data analysis and future experiments. Okay, first, intro. So here's some background. So first, how do plants use water? Plants take in water through their roots. They rely on, they use water to fuel photosynthesis, which produces sugars they need to survive. And they release excess water through pores in their leaves via transpiration. And plants rely on water to retain turgor, which is the strength of cell walls, which helps to keep plants growing upright. So a certain amount of water is always necessary for plant growth. And plants growing in soil can hold a certain amount of water until it becomes saturated, where any excess water will drain out of the soil, leaching nutrients in the process. So generally, there's a certain amount of water that's ideal for plant growth. And also overwatering can potentially kill plant roots, depleting by filling up pores and depleting the soil of oxygen, which roots require to survive, which we learned later. And changes in soil moisture, therefore impact ecosystems because plants rely on it. When plants are not surviving, that can have impacts throughout ecosystems. Also, plants can act to prevent flooding. And flooding is a big problem for humans as well. So soil moisture is important in agriculture too, making sure that our crops grow, and safety and preventing against flooding. And with climate change, changes in climate create a disruption to the water cycle, which is becoming more and more of a problem.
All right, so we decided to test what was interesting to us, and that was the soil moisture around the saltwater lagoon. For this, we developed a research question, and for those who don't know, a research question just helps us target what research and what data we gather when we're out and about, so we don't lose track of what we're doing. And our research question was, what is the effect of soil moisture content on the percent of plant cover? After that, we ended up creating our independent and dependent variable. Our independent variable is the moisture of soil within the area, measured in 1 to 10 units. And then our dependent variable, which was plant cover, and that was an estimated percentage. With that, we also have our hypothesis and null hypothesis. Our hypothesis was that soil with a high moisture content will yield a higher plant cover percentage. And our null hypothesis was that differences in soil moisture will not affect plant coverage percentage.
All right, so moving on to our experiment. Some of our materials. We used a quadrant, which is basically a square of PVC piping that allows you to estimate the amount of plant cover in a certain area. We used a conductivity measure, which basically estimates water levels by measuring how strongly the soil resists the flow of electricity between the two kind of stabby prongs. And then third, we used a tape measure just to measure about 5 meters in between each of our testing areas for each transect. All right, so our methods, we'd go to the very edge of the lagoon next to the water, start our measurements there, read the soil moisture there, the plant cover within the quadrant, and then we measure out 5 meters, walk back, and repeat 5 times per transect.
Okay, so now onto the results. So first, the evidence. So here you can see our graph, and on our x-axis, we have soil moisture reading from 0 to 10. Then we have plant coverage reading from 0 to 100%. And you can see here that a lot of our data falls to the left side of the graph. And the big red line running through our graph is a linear regression. And we use this linear regression to show us the general trend in our data. And with that, we also have a p-value. What a p-value is it shows the correlation between, and like how strong the correlation is between your two variables. And because our p-value was 0.05, it shows that we had a very strong correlation. Okay, so onto the discussion, the evaluation of our results. Basically, within our graph and within our linear regression, we can see that the general trend of our results is that as the soil got more and more moist, there was less plant coverage. The reason why we think that we got this is because what we talked about earlier was saturation, and how that sometimes waterlogged soil can mean that the soil doesn't, like the plants can't absorb the nutrients from the water because it keeps like running off the nutrients. So a bit about the area. So the area is the Rodale Lagoon, and this lagoon is basically like a lagoon and then the ocean, and then there's like a beach in between, and there's a sandbar, and the sandbar floods seasonally, which allows fresh water in, or seawater into the freshwater. And then recently the area has had kind of a drought, which has meant that the water hasn't been like flooding, and that kind of has dried out the area. And we saw that because a lot of our moisture reading, like as soon as we got off the brink of the water, was a reading of 1. And a bit with the big picture, basically what we can kind of tell from this is that there really is a healthy medium to soil moisture, and sometimes dry runs are a good thing.
Okay, so on to future research. We were wondering what in the world we could look at next. And so what we settled on was salinity of water, because Malcolm just mentioned that there is a combination of freshwater and seawater in the environment at a certain point. And so we were thinking that that's something really interesting that we could look for. And we would also have a bigger sample size for that if we had more time. And then our caveats for this that we could potentially solve for in a future research experiment could be that we didn't really walk into the reeds this time because we didn't have like the proper shoes and equipment to go in there and like come out clean. And so kind of having that equipment would enable us to get a bigger sample size and to get a bigger like coherent picture of the data.
Thank you.
Great work, everyone. We're going to go ahead and open it up to audience questions. So once again, if you have any questions in the live audience, we'd love to hear from you. Students, we'd love to hear any questions that you have. Please talk at the mic in the center of the room, but we'll start with questions from Morgan and myself, as Morgan was able to help them out during their field day. Morgan, do you want to start off with a question you have for this group?
Sure. I'm always interested in the challenges that people overcome when they are doing research. So does this group have any challenges or anything that was difficult as you were collecting this research?
Yeah. So originally our plan was to do about 5 transects, which is like basically a sector of 25 meters. And we realized very quickly that we were going to do a lot more than that, and kind of having to grapple with and find different places to take data from, because we kind of felt like, oh, like we went to all the places we could go without getting like super dirty. And so we tried to, like, we had to walk really far to find different places to take data from.
That's great. That was a great question too, Morgan. Do we have any in-class questions, Mr. Van?
Part of the grade is audience participation, which includes taking notes, but also asking questions. So, sorry, I didn't remind you, but if you could, but if they don't have any questions for our group, Yes, Kyler. And you might need to maybe come closer to the mic or closer to the computer. Thanks.
I saw one of the slides had like a caveat bullet point, but you guys didn't go into that quite as much. So I was wondering what caveats you had in your data collection.
Yeah, so when I talked about the caveat, that was kind of talking about how we didn't have as big of a sample size, because if you see the graph, most of our data points are in like the one soil moisture unit, because we couldn't go as close to the wet soil as we wanted. And so that is the reason that we could have gotten so much more data for drier soil. And so if we did the research in the future, then we would be able to wade into those reeds and get a bigger sample size.
It sounds like we need to invest in some multi-sized rubber boots for everyone to wade into the reeds and get more data. Does anyone else in the class have any questions, or anyone online have any questions they'd like to share with this crew?
We have two more,
Awesome.
Margaret and then Sam.
Kind of adding on to that, like, how do you think your data would have differed if you had been able to test like in the spots with a ton of reeds, or like walk into where it was muddier?
Well, the areas with reeds, they have reeds, had a much greater percent plant cover. So I think that would have influenced our data for the high moisture. The very wet soil we'd have more data points where that correlates with higher plant cover. And that could be like adaptation to salinity, like that could be related to saltiness in water. So maybe we need to go further in that research as well.
We'll take two more questions, then we'll move on to the next presentation. Sam and then.
So you guys have stuck a probe in the soil, right?
Yeah.
So what do you think would happen if you stuck it with more in the soil or with less in the soil, closer to the surface? How big does like the depth have results?
I feel like the depth throughout it was consistent, because I feel like sometimes we were able to get like a little bit more deep in, and sometimes it wasn't, but it always felt like really consistent. And I felt like if you stuck it deeper in, it was like going to read more wet or like less wet. So it kind of felt like it was really consistent throughout.
All right, and then one more question from Hannah, and then we'll move on to the insect group.
So like on the actual data set, there was one section where it was just like a bunch of dots in a row. Is there like a reason that way? Like there was just one, even though
Yeah, it kind of was just that, like how I talked about earlier, once you got off the brink of the water, the soil moisture like dropped significantly, like right away. And so even though like we kind of just kept moving further back, like right away it read at 1. And so like even though there was plants everywhere, it was still reading a soil moisture of 1, which we think is part of like the dry run that the area has been experiencing.
All right. Thank you very much, folks. Appreciate it.
Yeah, great job, you all. So I'm going to take the classroom down while they move on to the next crew who did research on insect biodiversity. And just do a few reminders that we're here at the Head Royce High School for their school program with us here at Headwater Science Institute. My name is Courtney Kudra, and I'm here with Katie and Morgan, who will also be presenting different groups that we were able to work with over the past 2 weeks. We had such an awesome time. We did some field work in the Marin Headlands. So it's been a lot of fun to work with them over the past 2 weeks. If you or your student really enjoyed this and would like to join our research experience, registration is currently open for this. It's a 12-week program where they can do their own research and get published. through Headwaters. So if you think that's something you or your student would be interested in, please check out that link. Otherwise, we are accepting questions and comments from the audience. So whatever platform you may be on, you should be able to add those if you have a question for your students. Otherwise, we're going to move on to our next group here who did theirs on insect biodiversity. I'm going to pull up their slides here. And Katie is going to go ahead and introduce this group that she was able to work with.
Sorry, got to unmute. Hi, everyone. I'm Katie. I'm the program director down in the Bay Area. And this was my first, not only my first group to work with, but my first project too. So that was super exciting. And our group worked on the insects biodiversity as they looked at the proximity to the lagoon. So I will let them take over and share the results they got.
I'm Haley.
I'm Simone. Oh, I'm Kelly.
And I'm Corbin, and this is our presentation on insect biodiversity.
Okay, so to start off with some background on the topic, the first question we have are why are insects and insect biodiversity important in the ecosystem? So first of all, insects maintain a healthy soil, they recycle nutrients, pollinate flowers and crops, and they control pests. They also are primary producers in the environment, and then they can serve as detritivores, which basically means that they can decompose waste and dead organic matter. And so all of that kind of makes them the foundation of the food web, and they stabilize the ecosystem. So right now, there's estimated to be about 5.5 million insects, but only about 1 million are actually classified and documented. However, there's been a rapid decline in insect biodiversity, estimated about 80%. Which means that this is a huge threat to the ecosystem because, as I mentioned, it's the foundation of the food web. And then to give a broad overview of what we did, we went to the Marin Headlands and we focused on measuring insect biodiversity in relation to the proximity from the lagoon. And we did this because we wanted to research why insect biodiversity has an impact on the environment and how it can be changed by certain habitats, which is why we also, you'll see later in this presentation that we included a graph to see how plants actually impact insect abundance.
So we had two research questions. The first one was what type of environment fosters the greatest insect biodiversity? And we measured insect biodiversity in terms of the number of insects rather than like species of insects. We did take note of species of insects, but we did our graphs with number just because it was hard for us to differentiate like completely between species. And then our second question was how does biodiversity of insects change as we move away from the lagoon? And as we moved away from the lagoon, we moved towards the ocean and the beach.
All right, so the independent variable for our experiment was our proximity to the lagoon, which we measured in yards. And then our dependent variable was the number of insects that were measured within the quadrant. This is mine too. Yeah. So the hypothesis for our experiment was that there will be more, greater insect biodiversity the closer to the lagoon because there will be more plants which the insects need to live for, laying eggs, shelter, resting, eating, and whatnot. And then the null hypothesis for this would be that proximity to the lagoon has no impact on insect biodiversity. Our materials that we used, we had the PVC quadrant, which we measured the number of insects within, and then we measured distance in yards using the measuring tape, 3 yards in between each measurement. Starting at 0 yards, which is like right next to the water line.
Here's our methods. So as Corbin said, we placed the quadrant 0 yards from the lagoon, right touching the water. Then we counted the number of insects, number of insect species, and the species of plants inside the quadrant for 1 minute. And then we measured 3 yards of distance from the back right corner of the quadrant, moving in a diagonal way. And then we set up the quadrant at this new location and repeated the process for 30 trials. And then after that, we repeated the entire thing starting at 0 yards from the lagoon, again, beginning from the opposite side of the bridge, just to get data from a slightly different environment.
And then these are a few more pictures of our methods, and this thing here is what we mean when we say 0 yards from it, just in case you're wondering what that looks like. And then here's a map of where we were going around with it. And then here's a picture of us after collecting data and moving.
Okay, so we have two graphs. This is our first one. On the x-axis, we measured the distance from the lagoon, and then on the y-axis is number of insects. As you can see, it shows a subtle negative association, meaning that as the distance from the lagoon increases, the number of insects decrease. And so we used a linear regression test to actually analyze this data, and we found that there's an R-squared value of 0.0483, indicating that there's only about a 4% of the data variability in this data is explained by the model. And what that basically means is that only about 4% of the variability in the dependent variable can be explained by the independent variable. So furthermore, a p-value of 0.09 also revealed that there's some confidence between the relationship of the distance to the lagoon and number of insects. And so it's not, we probably need more trials to actually strengthen this confidence, but using what we have, that's the best that we could do. Okay, so the second graph on the x-axis, we have 9 different plant environments, and then on the y-axis is number of insects again. And before I talk about this graph, as you'll see on the y-axis, it goes into negative values, and this is because the data classroom can't actually account for the nuances within the data, and so it can't like understand that insects can obviously not be negative. But anyways, we used 60 trials again for this data, and we used an ANOVA test to analyze it, and we found that the p-value is less than 0.01, which actually indicates an extremely strong confidence that the groups are different. And so that means that the different types of plant environments actually had a big impact on the number of insects and insect biodiversity.
Yeah, so to continue to go into more depth on the different plant species, we weren't really planning on measuring or accounting for different plant species as we came into the experiment, but we realized as we went on that there were a significantly different amount of bugs and insects surrounding different plants that we found, and we used the Seek app to identify different plants that we found, as you can see the screenshots above. So for example, we found European sea rocket, the top left, sea bindweed on the right, silver peachweed on the bottom left, and red sand verbena on the bottom right. These were just 4 of the many plant species that we found located on the beach, and we did find, as seen in the graph on the slide before, the significant difference between insect populations surrounding these different plants. And this could have something to do with the fact that, for example, I think it's, European sea rocket has flowers, so this could potentially attract bees as there is pollen and nectar in these flowers. So overall we did find a difference between where the bugs were located in relationship to these different plant species found in the lagoon, or near the lagoon. To move on to discussion, the first question that we had is our data conclusive enough to truly assess insect biodiversity in the Marin headlands? And to this we say, our data, even though we did do 2 trials of 30 trials, this is just a small picture, and we could always get more data to make our results more conclusive and really get a larger picture of the biodiversity of insects in this ecosystem. And our next question is, how much more data should we have acquired to have a more accurate understanding? And to this we say that maybe experimenting on different days during different seasons or even during different times of day could have furthered our understanding. And something that we did notice while we were experimenting on the beach on the particular day that we were there was that it was quite windy, and especially for the small flies that we were observing, this wasn't necessarily a good environment for them. So this windiness and the weather of that day could have impacted how many flies, for example, there were in the areas we were measuring on that day. Our next question was, did our results align with our hypothesis, and what conclusions can we draw? So yes, the trend line indicated in our first graph did highlight the decline in biodiversity the farther we got away from the water. So this does align with our hypothesis, so we therefore reject our null hypothesis. And even though, our data was pretty conclusive in showing that linear regression
that highlights the decline in insect biodiversity, which is what our conclusion or hypothesis was. And the next question is, what were the main patterns in our data? Like I've said many times now, we did find that there were more insects closer to the water, and we hypothesized that this was because there were more plant species closer to the water and overall more nutrients that the insects could feed upon and thrive nearby. And the last question is, why do we think we saw the results we did, and what caveats are apparent in our research? So as I mentioned before, the wind did kind of impact our findings, as flies could necessarily survive very well in these very windy conditions the day that we were there. We also found that in a lot of the areas we were measuring, there was just sand or a lack of plants. And we did find that if there was just sand or no plants, that this wasn't necessarily, there weren't that many insects living in areas where there was only sand and no plants. And finally, we want to account for human error, as we were trying to count different insects as they were flying by, and human error could be a component in this data that we received.
Part 2. What factors do we identify have the greatest influence on insect biodiversity? We found that plant species had the greatest impact, and as Simone said, there were a lot of bugs clustered around certain plants and not so many where there weren't plants or where there were dead plants. Distance from the water was also a factor, less significant, but still so. We found that there were a lot of insects closer to the pond water and less closer to the ocean water. So that speaks to the different kinds of sources of water, fresh versus not fresh. And then what could our results tell us about ecosystems like this one more broadly, especially when thinking about global environmental health? Our findings highlight the importance of habitat availability for insect biodiversity. In this case, the proximity to the water source or the lagoon or the ocean was very significant, which suggests that protecting and preserving habitats near a water source can promote insect biodiversity. Additionally, the observation that certain plant types attract more insects kind of shows that certain plants can be a biodiversity hotspot, and understanding which plant species are particularly attractive to insects can help in creating a healthy ecosystem where there's lots of different types of insects, and therefore increasing insect biodiversity. And maintaining and restoring habitats near water sources is not only beneficial for the insects, but also to the ecosystem in general, and even us humans, for reasons that Callie said in the introduction. Healthy ecosystems in turn contribute to global health and a healthy environment by promoting biodiversity, nutrient cycling, and ecosystem services that can help humans too.
Okay, so to quickly go over future research which could help strengthen our results. First of all, are there microhabitats within the proximity to the lagoon that support higher insect biodiversity? And what we kind of mean by this is, since we noticed that, as Haley said, there's certain plants that are kind of hotspots for insect biodiversity, how could you measure the why to this aspect and figure out like what is actually causing insects to be attracted to certain plants in comparison to others? The second question is, how does insect biodiversity connect to the larger species diversity? And as I said in the introduction, since insects are kind of the foundation of the food web, how does this actually impact the larger species diversity in the Marin headlands specifically, and then also just in general? And then how is insect biodiversity impacted by climate change? Since our results did show that the type of plants did impact insect biodiversity, if climate change is burning down trees and destroying environments of the habit of insects, then how will this actually affect the insect biodiversity and in turn the entire food web? And then how might a larger versus smaller sample lot size impact our data collection? Simone kind of talked about this. However, since we only measured 30 trials 2 times up the bank of going up to the beach, this doesn't really account for all the nuances in our data, because the plants were randomly distributed throughout that region, and so it's kind of harder to actually account for that, since we found that like there's a lot of insects in certain plants and then no insects when there's just sand. And so it would be more realistic if you could measure like say 1,000 trials and then do it all down the bank of the lagoon and kind of figure out how the insect biodiversity changes without having a difference because due to the plants. And then lastly, our last caveat is that we noticed in some big bushes of plants, it's really hard to actually see the insects in there. However, there are a lot, because when we accidentally would like kick the plant, a bunch of flies would come up, and so you know that there's a lot of insects within the plant that you can't see. So it's kind of harder to do this experiment when one, you can't classify the type of insects, and then two, you can't see a lot of them.
So yeah, finally we want to acknowledge all the people that helped us, and thank you to Katie, Courtney, and Mr. Van, and that's it. Thank you.
Great work, everyone. That was awesome. We'll start off just as a reminder, if anyone's in the audience wants to ask questions, please go ahead and share those on whatever platform you are on. We'll also take in class questions, of course, and I'll be writing those down and putting them online so everyone online can see them. But we will start off with Katie. Do you have a question for this crew and their really interesting research?
Yeah, what now, after having gone through this, would you do differently next time? It's always about fine-tuning your methods and all that. So what would you do differently if you were given the opportunity to do this again?
Oh, what did you say?
If we could do it again, probably the final method.
I mean, I also think that if we were to do it again, just collecting more data would be helpful. And then also, if we had a way to actually classify the insects, that would be good too.
I think also getting data from different like times might make it more accurate, because we did do it all back to back on the same day, like an especially windy day. Like different seasons, different days could have made it more accurate.
Yeah, awesome. And I do love how you guys did pivot. You were noticing that the plants became a focus for the insects, and I think that was great that as you guys were out there, you said, oh my gosh, these plants definitely have something to do with this. So that was a great pivot right in the middle of your research, which sometimes is hard to think about and to do, because you're set with one task and think you have to stick to it. But you guys did a great job, so well done.
Yeah.
We have one or two questions from the audience, Courtney, unless there's someone you got on the platform.
Let's go ahead and do some audience questions. We don't have any on the platform right now, so.
Okay. Sachi, do you want to go?
Yeah. How do you know that like the amount of insects in the plant or whatever, or how much you're measuring actually has anything to do with it? It's the diversity of insects, because isn't it just like the amount?
So yeah, to measure insect biodiversity, I believe we measure the combination of the number of different insects, but also the number of different species. And ideally, we would have liked to factor in the different type of insect species that we found. But because a lot of the insects were really small, it was hard for us to identify them through the Seek app that we were using. So ideally, we would have incorporated the different insect species into our data, but this wasn't really realistic with our method. Again, like ideally, we would have had this data, but it was just not possible with how we were doing our experiment.
So we'll do, trying to be mindful of time. We'll do 2 more questions. Jared and then Finch.
So if you were to do this again, like if you were to choose another variable to measure, what kind of 2 to 3 additional variables might you choose to measure?
I mean, yeah, I think that being able to classify the insects would definitely strengthen the kind of concept of insect biodiversity. So if we had someone who could actually kind of like see the small insects and then figure out what those were, that would be helpful.
Finch?
Y'all talked a good bit about how plant coverage impacts insects, like they need a place to stay and more water. Did you talk at all, did you think at all about the other way around? Like, for example, insects contributing to pollination, meaning more plants, meaning more places for them to live?
We didn't really think about that. We did keep track of bug species, and we didn't see many like bees. It was mostly detritivores, little crawly things. Not really things that are contributing to. I mean, they impact just not as directly as other bugs do. So we didn't really think about that. But
And how about we'll do one more, and then I know there are a couple of hands. I might just, I think, Anoki, you had your hand up. Just go ahead, Anoki.
Like, we talked about how important it is for what humans do to foster that. So a lot of insects, which is
I mean, I think it's kind of like a two-way street. You have to preserve the actual habitats of the insects, and then by doing that, you're also preserving insect life because you're taking care of the food they eat, and then in turn, they're able to give energy back to the environment by fertilizing plants and helping crops grow. So yeah, I think just overall, climate change is the biggest issue, and because of that, it's deteriorating the environments, and so being able to kind of give back to that would help.
Courtney, we have more questions, but I also want to acknowledge we need to make sure that the two presentations that are left have enough time. So if you all do have questions, if you could jot them down in your notes for me, that'd be great. And then I can that way I can acknowledge them that you had a question, just to get to ask it. So thank you, group. Thank you all very much for. Let's give another round of applause, and we'll have our next group come up.
Great work, everyone. That was super awesome. I know I learned a lot. I think our next piece for this group is getting them an on-hand entomologist. That would be awesome. So if any of y'all are looking for some volunteer opportunities and have that expertise, we could definitely use it. So thank you all so much as we get more folks lined up here. If your student really enjoyed this or is really interested in research in general, we do offer a research experience. This is the program that I run primarily. It's a 12-week program where you can get published with a scientific manuscript, develop your own research question, and get published through our Headwaters student journal. It's a really awesome option for someone who is interested in developing more science. But without further ado, we will invite our next crew, which I had the honor of working with at our field day in Marin Headlands. I know I had so much fun with this group. Not only were they adaptable, but they were super funny and really just not afraid to get their feet wet, literally, to collect some of this data. So thank you all for your really hard work during our field day, and I'm so excited to see your presentation. Y'all can go ahead and start.
Hi, I'm Margaret. I'm Hannah. I'm Salchi.
And I'm Audrey, and we decided to collect data on the effect of the proximity to the ocean and the road on water pH.
So for some introduction, we had the original goal of measuring the turbidity, so like how cloudy the water is. But then when we got to the field, the tool that you used to measure turbidity was unfortunately broken. So we had to do a last minute pivot, and we switched our dependent variable to the water pH, which we were interested in just because it has such a large impact on sustainability and how ecosystems thrive, which we'll talk about a little bit later. We conducted our research at the Rodeo Lagoon in the Marin Headlands. And just some quick background on Lagoon. That's a picture of the Marin Headlands, not specifically the lagoon. The lagoon was pretty much untouched by anyone and people until I think the 1930s, when the U.S. Army set up a base there. And then they built a road which reduced the lagoon to 80% of its original size. In lagoons, there's typically less nutrient cycling because they're pretty stagnant compared to the ocean. There's no waves. And then there's also, we researched that there's pretty frequent algal blooms in this specific lagoon during the summer where the pH increases. So that's definitely something that we took into account as we were researching and thinking about like the season that we were coming to Lagoon in. Yeah.
A little more background. So one thing that we took into account was the fact that paved asphalt concrete has a higher pH than like the neutral water. So that could have affected our results. Also, the survivability of plants, microorganisms, and sea creatures are very dependent on pH, and like increase and decrease of pH can lead to changes in the chemical states of pollutants, making them more harmful to the ecosystem in the water. Also, changes in pH can lead to more like toxic metals in the water and less bioavailability of nutrients for like plants to absorb. And also, the pH basically is optimal when it's between 6 to 8 for like everything in the water. And the last thing that we took into account was that ocean acidification can also cause a decrease in pH because CO2 it mixes with water and releases more H+ ions, which deteriorates shells in the water. So we wanted to see if that had any effect on the lagoon.
Okay, so our research question for this experiment was, what is the effect of proximity to the ocean and road on water pH? And our hypothesis was if we are further away from the ocean, the pH will be lower due to ocean movement near the lagoon cycling pollutants and nutrients, therefore stabilizing pH. And our null hypothesis would be if you're further from the ocean, the pH will not change.
Our independent variable was the proximity to the ocean, and we measured that every 50 meters. And our dependent variable was the pH of the water. So our materials, other than like us sacrificing our clothing and shoes to get deep in the mud, we used a 50-meter tape measure, pH meter, and paper and pencil to record our data.
And for our methods, we started at the beginning of the lagoon, like right near where the ocean met up with the entrance to the lagoon. We would dip the pH measure like Audrey's doing in that picture into the water, and we would record the data, and then we would measure out 50 meters further on the shore, and then we would repeat those same steps, and we did that all along the side closest to the ocean of the lagoon, and then we went to the other side closest to the road, and we repeated all of those steps. And I think altogether we collected 20 points of data, so 10 on each side. Wasn't the most, but it definitely took a lot of effort when we were getting dirty, getting our feet wet, and really committing to the process.
So here's just a little visualization of our oceanside sampling locations. So this is just a rough estimate of where we took all of our data points on the ocean side. And then here's another visualization of our roadside sampling locations. Again, just another rough visualization of where we took all of our data.
And for our evidence, this first graph is the distance from the ocean on the ocean side compared to the pH. And as you can see, the sample size is 10, so there's 10 data points. And the p-value was less than 0.01, which suggests that there's a strong correlation in the distance from the ocean on the ocean side and the pH. As you can see, the pH was kind of steadily decreasing as we got further from the ocean on the ocean side. And then for the road side, we have the same thing, distance from the ocean and pH. The sample size, again, was 10. And the p-value was 0.02, which suggests a pretty strong correlation. And as you can kind of tell from the graph, compared to this one, there are more outliers on the road side. So the correlation wasn't as strong, but you can still see a pretty strong trending downwards in the pH. And then we combine the two. So we have 20 data points on this one, and the pH and the p-value was less than 0.01, which, as I said, suggests a strong correlation in the distance from the ocean and the pH.
Okay, so in terms of results, we can obviously see from the graphs that in all of these cases, the pH lowered when we went further away from the water. And obviously we talked about like there are some outlying pH measurements. This could be due to a lot of things. It could be that like there's some other thing in that area impacting the pH. It could also just have been like a measurement error. There's a bunch of reasons why we can have outliers. But the basic idea is that like the decrease in pH was determined by that best fit line. And that best fit line may not take the outliers into account, but it still demonstrates that the pH decreases when we want further away from the water. This means that the hypothesis was proven correct, which means that we also rejected the null hypothesis. So just for some discussion, there's a couple of potential caveats. We had a pretty low sample size because each measurement took a lot of time. There's also some outlying pH measurements. Again, this could be caused by like a number of different factors in the area. So just like why this whole thing is important. Well, water could become unlivable for certain plants and animals away from the ocean if the pH changes too drastically, if it's not in that 6 to 8 like ideal state. And water may it's also worth noting that like water may naturally have a lower pH as we move away from the ocean, and like as we move away from the dependent variable, which is like moving away from the ocean. It's also really important that we recognize that like development and pollutants are dangerous for pH stability. Like pH must be stable for life because pH when it's outside of that 6 to 8 range can really mess with like chemicals and pollutants in the water and make them more dangerous and toxic, which is why like pH is a really important factor in terms of considering this study. So just for some further research, there's a couple more variables we could have tested. Obviously, we wanted to test turbidity, but we couldn't. There's also like velocity, which is water speed, and how that might impact cycling and like especially the cycling of nutrients and pollutants. We could have studied more locations. So we could, if we like had the capacity to move towards the center of the lagoon, which we did not, but if we did, that was also like another area of study. You could also do further research on the effects of pH on like the survivability of ocean life. So like if there is an area where pH is outside of that ideal range, what impact does that have on the life in that area? And we wanted to acknowledge Courtney Kudera and Mr. Van for the help on this project.
Well, that was such an awesome presentation. You all did such a great job. And I know if we tried to take samples from the center of the lagoon, you would be willing to swim out there. I know my shoes are still getting cleaned. So my question for you is, I know that there's a lot of other factors and variables you wanted to study, and due to time and equipment, weren't able to. But in that same vein, how do you think, if you were able to continue this project, what would your hypothesis be on the like relationship between pH and turbidity or velocity? You can choose either one, for the areas with the ocean side versus the road side.
I'm not sure if I'm interpreting your question right, but the road side and the ocean side, like we didn't like see much of we didn't see much of an effect of the road side onto the pH. And I think that like maybe next time we might change that. Like we might not take that as much into account as we did before, because I don't know if there's like pollutants actively being leached into the water from the roadside. Like it probably did happen when they were constructing the road, but that was a really, really long time ago. So I don't know if that affected it at all. So yeah, I don't know if that answers your question, but
no, that is a great answer. And it is a good thing to consider roads. Sometimes they have a big impact on things, and in your experiment, as you found out, it was more so the proximity to the ocean that had an impact on your pH levels. Now we can take any questions from the audience in the classroom or online. Once again, we're happy to take any questions from online, and I'm going to try to type any questions from the audience to post online. But we may not be able to hear you, so be sure to be close to that center camera and mic.
We have 3 questions, Courtney. First from Haley. And I'm going to ask folks if we could, we'll try to be brief and concise also, because we want to make sure our final group has a chunk of time.
You might not know the answer to this, if you were to study turbidity as a factor, how would you measure that?
We looked into like how you would measure turbidity. I think, yeah, did you explain it really well?
Yeah,
in our group.
so this is actually just Mr. Van explained to me. So I don't know if I'm gonna explain it right, but like I think it's something to do. It's like you take the water from like if the water's really cloudy, and then you take like purified water and you mix you add it into the you add the purified water into the
you add like drops.
You add drops
Yeah.
into the water
Yeah.
until it turns
Yes.
clear.
Yeah.
Well, or until it's just as cloudy.
Yeah,
as the
until
other.
they match, and then you can measure like that. So it's like you're comparing it basically to clear water.
Yeah, it's a standard drop solution. Yeah, that's right. Malcolm, and then Callie, if you could get a little closer.
So my question was, do you think that the pH values like on the like bank of the like water would match the like center of the water?
Not. Obviously, like there's more. The water's gonna get deeper as you move towards the center generally. So there's gonna be more like area for the water to like cycle through nutrients. It also there might be like different amounts of life or plants. Like that could also have an impact on pH. Sometimes plants will grow near the surface but not in the center because of the ability of sunlight to pass through the water, and so that might impact the pH differently.
It also to add, sorry, it also like there was areas we were walking through the water. We weren't exactly supposed to be there, but there was like thick layers of algae on there, and like literally even the temperature was different. So that might affect that because like sunlight obviously is warmer. It's like getting through to the water more in some certain areas than others, and that could like indicate like pH changes.
And Callie, last question. We'll have you guys want.
Yeah, you mentioned with the plants, but my question kind of has to do with, did you actually notice that there was like a difference in plant growth on like the roadside versus on the other side, since if the pH is lower, then that kind of make it easier for things to grow?
There was not really a difference in plant growth on the roadside versus the ocean side. There was actually like we had to walk through more plants on the roadside, so we like I don't think that had anything to do with like what we were measuring.
Thank you all very much. We're gonna have our final group coming up. Appreciate your guys' work.
Alrighty, we'll prep for our final group coming up here. Super excited for these folks to also be presenting to us today. They already have their slides up and seem to be ready to go. While their field partner, Devaney, is not able to attend today, I'm going to give them a brief introduction. These folks also were super adaptable and very fun to have in the field. They were able to collect a lot of different data in and around the lagoon. So I'm very excited to hand it over to them to give a presentation on their project.
Okay. Hi, my name is Kyler.
I'm Alex.
I'm Jared.
I'm Matt. I'm Sam.
And this is our Marin Headlands Research Project. Slide, please. So first of all, I want to start off with our research question, which is, what is the effect of pH on dissolved oxygen content around the shore of the Rodeo Lagoon? And so our hypothesis was that pH and DO were inversely correlated, meaning that a decrease in pH would result in an increase in DO. And our reasoning was that as CO2 increases, pH decreases as it becomes more acidic. As a result, more CO2 would result in more photosynthesis, which produces O2. So it'll be more O2. And our null hypothesis was that pH would not affect DO at all.
So what is pH? pH is a scale used to measure acidity or basicity of aqueous solutions. And as you can see here in this graphic, neutral is 7. It's a scale of 0 to 14, with numbers less than 7 being acidic and above 7 being alkaline. Essentially, pH is a measure of the concentration of H+ ions in solutions. So what higher pH and lower pH mean. Lower concentration of H+ ions and acidity in higher pH, and higher concentration of H+ ions and acidity in lower pH. So you might be asking yourself, what are the impacts of pH on the environment? Well, pH is actually essential to biological processes that can indicate chemical states or affect the chemical states of pollutants such as copper and ammonia, which can shift the exposure to these pollutants to different plants and organisms in the environment, thus changing species distribution.
So what factors affect pH? There are a lot, including rain, water seeping, which is like runoff just of water from like the grass, ground, coral, any organic materials. Chemicals, pollutants, and other contaminants too, which can come from humans as well. And this can make it either very acidic or alkaline, then also atmospheric carbon dioxide just from the atmosphere. And also, what is dissolved oxygen? It's a measure of how much oxygen is dissolved in the water or any body of water. And you can receive it from the atmosphere, aquatic plants, and running water. And typically a good DO for like the ocean is between 3 to 5 milligrams per liter.
Okay, so now getting to the test itself, our independent variable is pH, and our dependent variable was the dissolved oxygen content of a given area. Okay, so for materials and methods, one of our materials was using a pH meter, and we used a pH meter to measure the pH of water. And then we used a dissolved oxygen probe to determine the dissolved oxygen content. So those were our two kind of scientific tools. We held the tools in the water for approximately 10 seconds, recorded the data, and then used measuring tape for 15 feet and moved down the shoreline. So we repeated this about 47 times. And to mention a caveat, we took out the probe sometimes too early or too late. So that could potentially result in inaccurate data points.
And in addition, when we put the probes in, they were varying a lot. So we had to take an average rather than one set piece of data. So that might change it.
And also after each test, we'd have to pour water on our probes. And then the amount of water poured differed each time. And it could not sort of touch different parts of the sensor to clean them off. Okay, so for a bit of an illustration, our first step was measuring pH and DO. So here's Tyler placing the probes into the water at 3 different points, sort of varying in algae and plants. Step 2 is recording our data. We have Alex here writing down the data. We used Bluetooth, so it connected to a phone and he wrote it down. And then Alex and I measuring 5 meters to our next data point.
Okay, so analyzing data. So for our first graph, as we said with our variables, we had pH as our independent variable and DO as our dependent variable. And using linear regression, we saw that it seemed like there was a correlation between the variables. As pH increases, so it dissolved oxygen. But with another variable, amount of algae, we noticed that the amount of algae, specifically in the high areas, seemed to have some role in the data. So we see the turquoise dots seem to have high pH and high dissolved oxygen. So to investigate this, we made 2 new graphs, one where we renamed the amount of algae as a dependent variable and the pH as independent variable, amount of algae, pH as our dependent variable. And when we ran our data, we saw the p-value from low to high algae levels was 0.02, meaning that there's a very strong confidence that pH is increased with higher levels of algae. And then we did the same thing for another graph, just instead of pH, we have dissolved oxygen as our dependent variable. And this time our p-value was actually less than 0.01, meaning there's an extremely strong confidence that our dissolved oxygen is increased with higher levels of algae.
So now on to discussion. So first, let's go over some of the main patterns in our data. Firstly, dissolved oxygen generally increases with the pH. And then second, the amount of algae affects the results. A higher amount of algae results in a higher amount of pH and DO. And so how does this compare, how do our results compare with our hypothesis? So our hypothesis was that as pH decreased, DO would increase. As more CO2 was interacting with the water, so more photosynthesis would occur. However, our data showed that there is a direct correlation between pH and DO. As pH increases, DO increases as well. This actually makes much more sense than our original hypothesis because an increase in pH indicates that carbon dioxide is being taken out of the water, right? So plants are taking it up for photosynthesis and resulting in a higher DO as they photosynthesize. Of course, all this is only true under the assumption that the plants are producing more oxygen by photosynthesis than they're taking up by cellular respiration. And then this idea is further corroborated by the fact that when we had more algae in our data, there was also a higher DO and pH content as the algae photosynthesized.
Okay, so in order to, we wanted further information to kind of test the veracity of our claim. So then we looked at Charles Oster Chambers' 1912 paper entitled "The Relation of Algae to Dissolved Oxygen, Carbon Dioxide." This paper found conclusions in accordance with ours. A strong degree of alkalinity and an increase of oxygen from the 3 cc per liter to 7.8 when the algae was blooming, when the water was blooming with algae. So how does this affect sort of the Marin Headlands environment? The amount of algae is a potential factor of an increased DO and lowered CO2. So this can result in Park Services kind of using this knowledge and can limit or promote the growth of algae to maintain a homeostasis or equilibrium for the proliferation of aquatic creatures or plants.
So what? So let's talk about some of the environmental significance of pH in water. So one of these being ocean acidification. As you can see in this graphic, an increased amount of atmospheric CO2 causes a chain reaction, ultimately resulting in dissociation of H2CO3, leaving a high surplus of H+ ions in the water, thereby taking up some of the carbonate, which sea life requires to create calcium carbonate, which is shells, exoskeletons. So yeah, lower pH indicates higher levels of atmospheric CO2, and yeah, that's the impact of pH in water.
So DO levels are also very important because it basically indicates water quality. And you need DO to just sustain life in general. And animals in the ocean also require it. And we realize that a decrease in DO has a high correlation with algal blooms, which basically causes a lot more decomposition in the body of water and indicates a nutrient imbalance. And an increase in DO indicates an increase in primary productivity, which allows for either plants to do more photosynthesis, which allows for more energy in the ecosystem as a whole. So a couple future research questions we had were, how do different nutrient levels affect plant life, O2, and pH? And then, what are the ways humans impact DO levels in bodies of water? And how do humans impact plant life in bodies of water today? It's a bibliography. And
thank you for watching.
Question.
Awesome. What a great presentation, you all. I do want to be mindful of y'all's bell schedule. Mr. Van, do we have time to take a few questions for this group?
We do. We have 4 minutes.
Awesome. I'm just going to open it up to audience questions because I know that that is something your students need to do. So I'll give these 4 minutes to your students.
Corvin and then Sasha, if you all could come up a little closer, and if you didn't get to ask a question, we have time for one more. If okay, Simone, is it okay, Sasha? Thanks. And then if you don't get a chance, go ahead and write your question down, and then I'll be able to acknowledge the other question. Okay. All right, Corvin.
You said there was like some trouble with equipment. Do you think it's possible that that did anything with like student data at all?
Yeah. So there wasn't really trouble with the equipment. There wasn't necessarily trouble with the equipment. The equipment worked fine, but you'd have to place it in the water for a given amount of time because there was there's like varying data points, right? So it'd go from like 8 to like 12, right? So like sometimes we would take it out at 8 seconds or 12 seconds, not exactly always at 10. So there could be these very big jumps in data that were captured or were not captured based on the time at which we pulled each probe out.
And the problem we originally had with our equipment was that it wasn't connecting to the phone. So it wasn't actually like the ability of the probe to measure it. It was just whether or not we could get it to actually connect to Devaney's phone.
Yeah, so you found like a big impact, algae having a big impact on dissolved oxygen and pH. Like considering that the lagoon has brackish water and floods occasionally, like how do you have any idea of how far algae could grow, or like how much that would be able to grow considering the flooding from the ocean?
I think that'd be a great place for further research. We didn't actually get, because algae wasn't intentionally originally wasn't a part of our experiment. We only realized it as it was a correlation with how much plant growth there was. So I think that'd be a great place for further research as we learn about algae growth.
Thanks.
I was just going to ask what you think the data would look like if instead of measuring just distance down the lagoon, if instead you measured distance from the algae specifically.
And in kind of a way we did do that, right? Because think about it, if we're going towards algae on one side, we'd also be leading on the other side. So we kind of did do that.
All right. Courtney, were there one more question out there from the virtual audience or?
We don't have any questions from the virtual audience right now, but I have a question for this group. I think it was super interesting, your project that you were able to do here. And my question is, how do you think the different algae growth and dissolved oxygen levels, if you were to measure a variable other than pH, what would be your third variable that you think would be impacted based on those two, dissolved oxygen and the algae?
I think we talked a little bit about salinity because that would impact plant growth, and also it would cause different kinds of life to be prevalent, like sea life to be prevalent in the lagoon. So that could obviously change how much respiration is happening, how much photosynthesis is happening, and then there might even be some chemical aspects to that as well.
Great. Well, that was awesome. Thank you all so much for your awesome presentation. I'm going to go ahead and take you down here so you all can adhere to your bell schedule. But everyone here on the live stream, thank you so much for joining us. We really enjoyed working with Mr. Van's environmental science class. The past 2 weeks, they had some awesome presentations, and thank you for coming and watching today. We also do have a few other things that Headwaters offers. We have the research experience, which is a 12-week experience that individual students can participate in and write a manuscript for publication. We also have these school programs that run in the Bay Area and in the Truckee-Reno area, or various areas throughout California and parts of Nevada. There's a lot of things available on our website. So we're so excited to be involved with your students and your schools. So thank you all for coming, and we look forward to seeing you in the future.